Kingston.
Good afternoon, participants, and good morning to our guests for this webinar. Our topic for this afternoon is lifelong learning and the future of work, focusing on current trends and opportunities in drug discovery, European perspective. I call upon Professor Shivakumar, the head of the Institute, Geetham School of Pharmacy, Hyderabad, to deliver his welcome address.
Yeah. Thank you, Kingston. I would like to welcome our today's guest, Dr. Hari, who has been accepted to deliver his lecture. And thanks to Kingston, he is the one who has organized to share his colleague or friend from UK to have the discussions today. Welcome all the participants to the event. I believe that you'll have a great insights to listen from Dr. Hari to share with. Dr. Hari has got a lot of experience with the industry, basically with the R&D, research and development. I hope you could, staying far from UK, it's a very good challenge for us to learn and understand what are the perspectives which are at the global level. I think Dr. Hari would be, the platform is all for Hari to share his experiences with all of us. It would be a great opportunity. Yeah, thanks, Dr. Hari, and also Kingston and all the participants. I hope the session would be a great learning session for all of us. Thank you, Kingston. Yeah, over to you.
Thank you, Professor Shivakumar. It's my pleasure to introduce our guest today, Dr. Hariharasudhan Bose, Discovery Program Lead at GW Pharmaceuticals, England. Dr. Hari started his pharmacy career in the year 1999 at JSS School of Pharmacy, UT. He has done his master's in pharmacy practice and his doctorate in the development of novel sample extraction techniques to address the challenges faced in LCMS-based bioanalytical methods and their validation. He began his job career as bioanalytical scientist in India. Later, he moved to United Kingdom and began his career in LGC Groups in the year 2010. Now being a discovery program lead in GW Pharmaceuticals, he is responsible for monitoring, tracking, management, and review of discovery programs, timelines, and budgets that aims to progress the company's research and development strategy. With that, I'm inviting Dr. Hari to deliver his talk.
Thank you. Thank you, Dr. Kingston, and thank you, Dr. Shivakumar, for inviting me to give the presentation. It's my pleasure to share my knowledge and experience with the students. And for all the attendees, nice to meet you all. I hope I can start the presentation. Am I audible to all of you?
Yes, Hari, you can go ahead.
Okay, cheers. If you have any questions in between the presentation, you can hold on and ask the questions at the end of the presentation. But I don't mind even being asked in between. Let me share. Okay. So today we're going to talk about the current trends and opportunities in drug discovery. As an introduction, what I would say is we will just look into what is discovery, and then we will look at the current trends, and we can talk about the opportunities, how we can involve and get into the discovery field. Okay, so in the past, most drugs have been discovered either by identifying the active ingredients from traditional remedies, like the herbal drugs, or by serendipitous discovery. Serendipitous means finding something accidental when we do something else, or when we work on some other molecule which would be useful for other indications. That's how they found the molecule discovery molecules. So example of that serendipity action is minoxidil, which was developed originally for hypertension. Then later it converted to be a drug to hair growth. So that's how the serendipity action is there in the discovery field. And now we know diseases are controlled at molecular and physiological level. Also, shape of a molecule at an atomic level is also understood, and we know the hands of information on human genome. So by all these means, we can specifically design a drug and which will be specifically targeted to a disease. So there are a few methodologies. Still, this is applicable all over the world for drug discovery. This is random screening. Just identify certain diseases and screen certain drug molecules which we think that could be a suitable remedy. So we do screening randomly, or we do molecular manipulation. We have taken some molecules which would be already in use for a disease. We can manipulate it to make it more efficient or make it less metabolizable. That kind of activity changes can be done by manipulation. And the next one is molecular designing. By use of the medicinal chemistry, we can specifically design a molecule to suit a drug target. That means the protein which is there in the body, which is responsible for the diseases. So we can design certain molecules to fit into that receptors, and then we can give the action. So that's use of the medicinal chemistry. And the drug metabolites. So mostly we all know after taking the drug will be metabolized, and those metabolites only cause the effect. Sometimes the product itself will be an active drug, but mostly the metabolites. So what we can do is specifically synthesize those metabolites so that the efficiency can be increased. We can reduce the level of the dose. And as of course, as I said, the serendipity action, that's another method of drug discovery. So there is a huge timeline for any drug discovery and development program. So it takes approximately 15 years and $2.5 billion US for one drug to come into the market. So when it during the discovery stage, if we scan around or 10,000 compounds, that will only come up to 250 compounds when it goes to the development stage, from the target identification and to the lead optimization. When it goes to the preclinical stage, animal testing, it will be only 250 compounds. From there, only 5 compounds can reach to the clinical trials. From there, out of that 5 compounds, only 1 compound could be approved by either FDA, MHRA, or other regulatory bodies. By looking at this, the huge investment and the timeline, you can imagine how much volume of work is put into drug discovery. So there are different stages in drug discovery. The first one is called the target selection, which includes target identification and target validation, which is done by certain methods like cellular and genetic target identification, genomics, proteomics, and bioinformatics. And then the next stage is called lead discovery. This is nothing but lead identification and lead optimization. So the first one, target identification, is identifying the protein which is responsible for that disease, the receptor cellular level. That's mainly the pathology of the disease. Lead identification is responsible, is mainly concerned with the identification of the molecule which act on that target. So that's lead identification and lead optimization. That comes in the lead discovery, where the synthesis and isolation from any herbal drug or computational chemistry is used to identify a lead compound, and assay development to screen that, and then high throughput screening. High throughput screening is nothing but having a volume of chemicals, and that will be run against a proof of concept methods with that particular target, either in vitro or in vivo. And then by that way, we can select some hit compounds. They would be promising lead compounds. They are the compounds will be taken out from the high throughput screening. That's called high throughput screening. Then the medicinal chemistry. Here we would develop a library of compounds. After the hit high throughput screening, we'll have hundreds and thousands of compounds. We will have to choose those compounds and keep a library which would be a potential drug. So that will be done based on the SAR activities, structural activity relationship studies, and in silico screening using the data science and chemical synthesis. Then it goes to the in vitro study, where the ADME experiments are done, like how it's absorbed, distributed, metabolized, and eliminated from the body, and how it is, what is the affinity towards the drug target, and how the disease modification is happening, what is the mechanism of action. And by doing all these studies, we can come to a conclusion where the lead candidate can be taken, that can be refined. Then these lead compounds go to the in vivo studies after the in vitro studies, where the animal disease model will be developed, where based on the target, which protein, and which kind of a disease pathology state is there. That kind of a disease model developed in the rat or rabbit or any animal. So if it is diabetes, we will generate a diabetes in a rat. So we'll remove the pancreas and generate the diabetes. We will generate that disease model in animal. And then we inject the drug and find the efficacy. And that pharmacological study will be there, and admin study will be there in animal. And then it goes to the human later stage. And behavioral studies, when we induce the drug, what is the behavioral changes to the animal, that has to be tested. And functional imaging, after the drug administration, all the tissues are sliced, and will go to the imaging, where they will study what is the distribution, how that affected, and what is the pathological state of the tissues. That will be studied. And then it goes to the ex vivo studies. Ex vivo, nothing but the pathological studies. Then it moves to the clinical trial. You all know what's clinical trial. It's the study in humans. Okay, so now target selection. So this is the first stage in any drug discovery. Target selection in drug discovery is defined as the decision to focus on finding an agent with a particular biological action that is anticipated to have therapeutic utility. It is influenced by a complex balance of scientific, medical, and strategic considerations. Target identification to identify a molecule targets that are involved in the disease progression. Like I said, the protein, which is responsible for the disease. Target validation to prove that manipulating the molecule target can provide therapeutic benefits. So by doing some action, increasing the action potential, changing the receptor, or manipulating that target protein, we can change the disease condition of that cell. So that is target validation, just proving that this is the actual target. When we modify this target, we can definitely achieve the cure. So there are a few different classes of targets, which I would say mainly the receptors. The 45% time it will be G-coupled protein receptors. Sometimes it will be enzymes. Sometimes it could be hormones or the factors in the blood, or the ion channels and nuclear receptors. There are a few techniques to identify these. That's called cellular and genetic targets, so which involves the identification of the function of a potential therapeutic drug target and its role in the disease process. For small molecule drugs, this step is the process involves identification of the target receptors or enzymes, whereas for some biological approaches, the focus is at the gene or transcription level. Drugs usually act on either cellular or genetic chemicals in the body, known as the targets, which are believed to be associated with the disease. So scientists use a variety of techniques to identify and isolate individual targets to learn more about their functions and how they influence diseases. Nowadays, we have a technique called crystallography. We crystallize the receptor separately and identify the structure of that receptor or that protein. So protein crystallography is a newer technique which involved studying that target. And compounds are then identified that have various interactions with the drug target that might be helpful in treatment of the specific disease. Then based on that structure of the target, structure of the protein, we can identify a few chemicals. So we know what the functional groups in a chemical and how it can be connected with that target. That's how we develop a lead compound. Then it's genomics, the study of genes and their function. Genomics aims to understand the structure of the genome, including the mapping of gene and sequencing the DNA. Seeks to exploit the findings from the sequencing of the human and other genomes to find new drug targets. Human genomes consist of a sequence of around 3 billion nucleotides. So, you know, adenine, guanine, and thymine, all these proteins, which in turn probably encode 35,000 to 50,000 genes. There is an African-American scientist, Drew. He estimates that the number of genes implicated in disease, both those due to defects in single genes and those arising from a combination of genes, is about 1,000. Based on 5 or 10 linked proteins per gene, he proposes that the number of potential drug targets may lie between 5,000 and 10,000. Single nucleotide polymorphism, which is like changing one protein in the gene, are used to compare the genomes from both healthy and sick people and to identify where their genomes vary. Proteomics, this is another technique to identify the targets. It is the study of the proteome, the complete set of proteins produced by a species, using a technology of large-scale protein separation and identification techniques. It is becoming increasingly evident that the complexity of biological systems lies at the level of the protein, and that Genomics alone will not suffice to understand these systems. So just studying the genome will not be helpful. We'll have to understand how the RNA is generating the proteins and how the protein is evolving in the body. So studying the proteins is also important, equally important as studying the genomes. It is also the protein level that disease processes becoming manifest and at which most 91% drugs act. Therefore, the analysis of proteins, including protein-protein, protein nucleic acid, and protein ligand interactions, will be at most importance to target discovery. Proteomics is the systematic high throughput separation and characterization of proteins within the biological system. Target identification with proteomics is performed by comparing the protein expression levels in normal and diseased tissues. So 2D page is a separation technique used to separate the proteins, which are subsequently identified and fully characterized using LC-MS, liquid chromatography, mass spectrometry, and mass spectrometry. That is the triple code mass. Bioinformatics, this is another computational technique. So bioinformatics is a branch of molecular biology that involves the extensive analysis of biological data using computers for the purpose of enhancing biological research. It plays a key role in various stages of the drug process, including target identification, computer screening of chemical compounds, which is the in silico method for lead identification, and pharmacogenomics. Pharmacogenomics is nothing but the genomic effect and pharmacological activity, which is mainly used in the personalized medicine. Bioinformatics methods are used to transform the raw sequence into meaningful information, genes and their encoded proteins, and to compare whole genomics. Can compare the entire genome of pathogenic and non-pathogenic strains of a microbe and identify gene proteins associated with the pathogenomes. Using gene expression microarray and gene chip technologies, a single device can be used to evaluate and compare the expression of up to 20,000 genes of healthy and diseased individual at one time. So next, after the target identification using those techniques, we are moving into the field where lead discovery. So in the target identification, we now know the structure of the protein, which is responsible for disease. Now we'll have to find a compound which suits or combined with that target to produce the effect. This is called lead discovery. Lead discovery is also two parts. One is lead identification and lead optimization. Identification of small molecule modulators of protein function, the process of transforming these into high content lead series. Synthesis and isolation, separation of mixture. So these compounds can be separated from a mixture of compound or from a herbal drug, or separation of impurities. And then after separating, the impurities has to be removed, and either it could be synthetic or botanical. We'll have to get rid of the impurities to separate the compounds which would be potential to the drug targets. And in vitro chemical synthesis and biosynthetic intermediates. These are all the ways to separate and bring out those lead compounds. So combinatorial chemistry, rapid synthesis of or computer simulation of large number of different but structurally related molecules. Search new leads, optimization of target affinity and selectivity, ADME properties, reduce toxicity and eliminate side effects. These all can be done with the use of chemicals. Not only synthesizing large quantities, but by identifying the structure and putting them, analyzing the data science, we can compare those previous compounds and their ADME properties is already stored. Using the data science, we can filter and find the compounds which are more suitable in our body, which would be more suitable to the pharmacokinetic effects. Assay development. So after identifying few set of compounds, we know which would be structurally related compounds. That we have to develop assays. Number one, to evaluate them. Number one, to evaluate in the animal model, and also to quantify them using an instrument. So expressed protein targets or enzyme substrate interactions. High throughput screening. As I said, there will be hundreds and thousands of compounds we would have synthesized and assay developed, and we would have done an in silico study. Then this has to be tested with the target. That's called high throughput screening. Screening of the drug targets against selection of chemicals. So we have these chemicals, and there will be a method for proof of concept, either in vitro or in silico, and then we run through them and find which would be more suitable to that target. That is called high throughput screening. Identification of highly target specific compounds. Then we go into the field of medicinal chemistry, where we alter the compound, whatever the promising lead compound, that will be altered and made it more suitable to the drug target. Okay, it's a discipline at the intersection of the synthetic, organic chemistry, and pharmacology. Focuses on small organic molecules and not on biologics and inorganic compounds used in drug discovery to identify the hits. Hits is nothing but the compounds coming out of the lead, the high throughput screening. Then lead optimization with to lead, and we'll have to choose which will be more suitable. So that will be the lead compound. And process chemistry and development, then that has to go to the chemistry department and develop it into the way we want it. So library development, collection of stored chemicals along with associated database. As I said earlier, after the medicinal chemistry, within the medicinal chemistry, after the lead discovery, we take a certain number of compounds, we prepare them as a library. So they'll be in a series based on their structure. And then which assists in high throughput screening. Again, we will be running through the high throughput and helps in screening of the drug target. So we can get the hit compounds from these library compounds based on organic chemistry. So this is a combination of organic and pharmacological studies. The next stage is lead optimization and medicinal chemistry involvement is also the heavy involvement of structural activity relationship. So this helps to identify the pharmacophore. So pharmacophore is the precise section of the molecule that is responsible for the biological activity. Enables to prepare more active compound, allow elimination of excessive functionality. Then the in silico screening, as I said, we have extensively already used the computational technique to select the chemicals. Now we should use the data science after the lead optimization. The selected compounds will be run through this in silico screening. So computer stimulated screening of chemicals and helps in finding structures that are most likely to be bind to the drug. Filter enormous chemical spaces. We'll have to filter large number of chemicals are filtered in here. And this is more economic than just running with any pharmacological studies like using the high throughput screening. So now the chemical synthesis. So after all the modification and manipulation of the lead compound, which involve we move into the chemical synthesis, so which involves production of lead compound in suitable quantity and quality to allow large-scale animal and eventually extensive human clinical trials. So this is the stage where we have identified or optimized a lead compound, and we synthesize the compound in a large quantity. Now we can use those quantity into the animal study. Optimization of chemical route for bulk and industrial production. So there are different routes to synthesize them, so that has to be validated and optimized. And then it should be made suitable for formulation. Now the formulation department comes in role. So the druggability of the compound has to be decided, and which form, either it's liquid form or in emulsion or ointment or whatever the form is, we'll have to decide in this stage. Then we move on to the in vitro studies. Before getting into the animal, we'll have to, because of the cost effectiveness, we'll have to study them in vitro. So studies using a component of organism, test tube experiments it's called. For example, cells derived from the multicellular organism, subcellular compounds, ribosomes and mitochondria, cellular, subcellular extracts, and purified molecules like DNA and RNA. We even use the cadavers, liver and other tissue cells to prepare our in vitro platforms. So advantages: studies can be completed in a short period of time, reduces the risk in post-clinical trials, permits an enormous level of simplification of the system, because in vitro, as I said, we prepare the tissue cells from the human after the in the dead body, from the dead body. So that's why it mimics the clinical trials. It's not only, sometimes when you do studies in animal and when it goes to clinical, human metabolism will be totally different for few compounds, and then it totally stops the compound in progressing. So that kind of a risk is avoided when we do the in vitro studies. Permits an enormous level of simplification of the system. Investigator can focus on a small number of compounds. So we know these are the compounds which will work in humans. So we can just focus on those compounds to get into the animal study. So by doing that, we can reduce the cost as well. Drug affinity and selectivity. So this is also important. Even though we have chosen the correct structure, we should know how much affinity it has towards the target. So drug affinity is the ability of the drug to bind to its biological target. it could be receptor, enzyme transport system, or etc. Selectivity, drug should bind to specific receptor site on the cell. So there should be also specificity. It can't just go and bind to any receptor in that cell. Cell disease models. So in our isogenic human disease models, or a family of cells that are selected or engineered to accurately model the genetics of a specific patient population, in which, as I said earlier, so we know what are the specific patient population. We can just mimic cell, the petri dish. We can create the cells in vitro using our cell disease models. So isogenic human disease models. Stem cell disease models. So adult or embryonic stem cells or carrying or induced to carry defective genes can be investigated in vitro to understand the latent molecular mechanism and disease characteristics. So there's one slide missing for mechanism of action. You all know what is the mechanism of action. So just understanding the action of the drug, it's like getting into the molecular pharmacology, which receptor it is activating. After activating, what are the potentials are initiated and how this is influencing the nucleus. So understanding that pattern is called mechanism of action. Then the lead candidate refinement. Optimizing chemical hits for clinical trial is commonly referred to as lead optimization. The refinement in structure is necessary in order to improve potency, oral availability, selectivity, pharmacokinetic properties, and safety. As I said, we know the candidate is working in that target, but we know it's working with the human in vitro studies, but we should improve the druggability and usefulness of the compound, because the potency has to be increased so that the dose given to the patient will be less. And the oral route has more patient compliance. So therefore, we'll have to develop which is more suitable to give orally. Otherwise, if you give parentally or the other ways of ingestion can be less, can potentially lead to a less patient compliance. And we'll have to be more selective as well. As I said, it has to go and bind to the particular receptor or the particular target. And we have to improve the pharmacokinetic properties also to the drug, and safety profile again. So for that, we need to do some modification and optimization. That's, again, it's a part of medicinal chemistry that can be done simultaneously with the in vitro studies, because during the medicinal chemistry stage, we just sort of design the compound to bind to the drug. And then we move on to the in vitro and we find there the compound is working with the human in genomic or the microbe in vitro studies. When it is working, then we'll have to improve the bioavailability. And for that, we have to do certain other modifications by changing the functional groups or tweaking the compound in certain ways. Then we move on to the in vivo studies. That's called preclinical studies. So there are n number of preclinical studies based on the drug target, on the drug. We have different animal models. It could be rat, rabbit, mouse, monkey. But for at least 2 of the animal models should be done for any particular drug. So this experimentation using a whole living organism gives information about metabolic profile, toxicology, and drug interaction. Toxicological study is also very important. After your proof of concept and pharmacological effect study, we give the drug up to the lethal dose, and then after the lethal dose, the drug is the animal is dissected, and the drug distribution and what are the tissue damages has to be extensively studied separately. Animal models of the disease states. Test conditions involving induced disease or injury similar to human conditions. That's called the animal model by inducing a disease condition or injury. Must be equivalent in mechanism of cause. Can predict human toxicity in 71% of the cases. So SID mice, HIV, NOD mice, diabetes, and dinoreo gene function, these are the examples of animal models. Then not only the disease models, we'll have to go into the behavioral model also. This is more applicable to the neuropsychological drugs. But having said that, it can be done in other drug targets also, like for cardiovascular or any other things, because of the potential side effects. So tools to investigate behavioral results of the drugs used to observe the depression and mental disorders, how self-esteem and suicidability are hard to induce. So suicidability is another condition which could be a side effect for a few drugs by increasing the serotonin, 5-HT serotonin level in the brain. But this disease model, it's harder to induce that in animal model, because animals have less tendency to have that neurotransmitters induced. And despair based on forced swimming and tail suspension, reward based, anxiety based. These are a few examples of the behavioral model. Functional imaging, a method of detecting or measuring the changes in the metabolism, blood flow, regional chemical composition, and absorption. There are some techniques we can slash the animal bodies, and using the radio label compound, we could have traced the compounds using imaging studies. That's what this is a mandatory regulatory requirement now. After your, I mean, in addition to the toxicology, toxicology studies and behavioral studies, we'll have to do the imaging study also before getting into the human trials. Traces or probes used, metabolites used, MRI scanning, CT scanning, all the few scanning techniques. There are few advanced scanning techniques is also available in the Western countries now. Ex vivo studies, experimentation on tissue in an artificial environment outside the organism with a minimum alteration of a natural condition. That's called ex vivo studies. It's similar to in vitro studies, but in a tissue organs, but they are live, which could be artificial as well. Counters ethical issues. Example, measurement of tissue properties and realistic models of surgery. So, now after all these clearance from the toxicology, in vivo pharmacology, in vitro, and gene tox, all these activities, now the drug can be filed for the human trials. So that's called clinical trials. Set of procedures in medical research and drug development to study the safety and efficacy of a new drug, essential to get marketing approval from regulatory authorities, may require up to 7 years. So this is a new trend, a trend called phase 0. Usually we always have phase 1 to phase 4 clinical trials. This phase 0 is called the microdosing. This is a newer implementation. So recent designation, also known as human microdosing studies, first in human trials conducted to study exploratory investigational new drugs. So before giving to any volunteers, 1/10 of the compound strength. So if you are giving a paracetamol for 500 mix, so you can reduce to 1/10, like 50 mg of paracetamol can be given to humans and see what is happening. Then we can escalate to the actual dose to a normal volunteers. That's into the phase 1. Designed to do speed up the development of promising drugs, concerned with preliminary data on the drugs, pharmacodynamics and pharmacokinetic data, efficacy of preclinical studies, which can prove the efficacy of the preclinical studies. So now after doing the phase 0, we can get into the phase 1 study. Clinical pharmacologic evaluation. First, it's called clinical pharmacology. The first stage of testing in human subjects, 20 to 50 healthy volunteers are involved in the study, concerned with human toxicity, just to study with the normal dose, is there any side effects or toxicity? Tolerated dosage range, whether it is being tolerated or not in humans, and pharmacology and pharmacokinetics. It should be pharmacokinetics, not dynamics. Phase 2, controlled clinical evaluation, where 50 to 300 patients. So after evaluating the safety in the normal volunteers, not patients, now we are moving into the patients for 50 to 300 patients. Controlled single blind technique, concerned with safety, efficacy, and drug toxicity, and also drug interaction and efficacy studied here. Then we are moving into phase 3 study, extended clinical trials, most expensive and time consuming, 250 to 1,000 patients should be involved in. Controlled double-blind techniques, concerned with safety, efficacy, comparison with other drugs, and package insert. Now the phase 4, after the phase 3, the drug is marketed. It's been, it should be approved by the regulatory authorities. So after the phase 3 and the drug into the market, we will do the post-marketing surveillance. Mainly the pharmacovigilance studies comes after the phase 3, which is into phase 4. Designed to detect any rare or long-term adverse effects, adverse drug reaction monitoring, and pharmacovigilance. So these are the drug discovery. Now you have seen how the drug is developed right from the chemical and target identification up to the clinical stage and approval, and then pharmacovigilance studies. Now we are moving into the second part of our presentation. So what are the current trends in the drug discovery? I didn't cover the extensive trends in this presentation, because it might be too vast and overwhelming. I've just captured only 3 aspects which are more relevant and at this current stage, and they are extensively used in the Western countries. So you can focus on those areas when you want to do any specialization, which has more scope. That's why I covered these 3 things. So obviously, if I don't touch artificial intelligence, it will not fulfill the presentation, because artificial intelligence is the core and current affairs everywhere, not only in the medical research, everywhere, clinical, whatever, coding, anything you touch, it's artificial intelligence now. So it didn't stop there, it come to drug discovery as well. Traditional drug discovery methods are target driven. Example, known target is used to screen for small molecules that either interact with it or affect its functionality in cells. These approaches work well for easily druggable targets that have a well-defined structures and whose interactions inside the cell are understood in detail. However, these methods are extremely limited due to the complex nature of the cellular interaction, as well as the limited knowledge of the intricate cellular pathways. So now the AI is involved in the drug discovery. This can overcome these challenges by identifying novel interactions and inferring functional importance of different components of a cellular pathway. AI utilizes complex algorithms and machine learning to extract meaningful information from a large data set. Data set of RNA sequencing can be used to identify genes whose expression correlates with a given cellular condition. So we can involve this one in target identification by studying the RNA sequencing. AI can also be used to identify compounds that could bind to undruggable targets, example, proteins whose structures are not defined. As I said, we have crystallographic techniques to define a receptor or a protein, but that's not possible for all the proteins. So there are some unidentifiable proteins in our body. So we can even involve the AI to those kind of proteins and those kind of peculiar diseases where we don't have any drug now. Through iterative simulations of interactions of different compounds with small pieces of a protein, a predictive set of compounds can be easily identified in a relatively small amount of time. That's, time is a biggest factor in this artificial intelligence techniques. So traditional versus artificial intelligence-based drug discovery methods. The traditional method, target-driven, work well for easily druggable targets that have a well-defined structure and whose interaction inside the cell are understood in detail. So only if we know the signaling pathway, it's easy to do in traditional way. Extremely limited due to the complex nature of the cellular interaction. Cellular interaction molecular pharmacology is always complex, and we can't exactly predict. It's all assumption work as of now. We don't know exactly what's happening in a live cell. And limited knowledge of intricate cellular pathways. When it comes to AI-based, data-driven, so this is completely data science. Complex algorithms and machine learning can extract meaningful information from a large data set, identify compounds that could bind to undruggable targets, proteins whose structures are not defined. Benefits of applying the artificial intelligence to drug discovery. The application of AI to drug discovery has the potential to revolutionize the current time scale and scope of the drug discovery. AI does not rely on predetermined targets for drug discovery. Therefore, subjective bias and existing knowledge is not a factor in this drug development process. AI utilizes the latest advances in biology and computing to develop state-of-the-art algorithms for drug discovery. With a rapid increase in processing power and reduction in processing cost, AI has the potential to level the playing field in drug development. AI has a higher predictive power to define meaningful interactions in a drug screen. Therefore, the potential for false positive can be reduced by carefully designing the parameters of the assay in question. Most importantly, AI has the potential to move drug screening from the bench to a virtual lab, where results of a screen can be obtained with greater speed, and promising targets can be shortlisted without the need for extensive experimental input and manpower hours. So the main thing is we are not working on the wet lab, doing all these experiments and taking so long time and manpower involvement. We can just sit in the computer and do our initial screening shortlisting. That's the main advantage of the AI. Drawbacks of applying AI to drug discovery, as this is the case with any advance that brings a paradigm shift in our understanding of an existing technology, AI still cannot replace a human scientist entirely in the process of drug discovery. AI predictions are as good as the algorithms used to investigate the data set. So if you have a good algorithm, the predictions could be better. But the algorithm, mainly based on the algorithm is nothing but the data we fed into the computer previously. So the data fed is limited because every pharmaceutical company have their own data. They don't feed into the public algorithms or public. It's not widely available because it's all patented and it's all secret. So we have limited data sets available publicly. So that means if the algorithm is not good, the prediction is also not good. The algorithm should clearly lay out the criteria that should be used to parse out meaningful information when the results are in the gray zone of interpretation. AI can suffer from algorithm bias, whereas the creator's own bias manifests itself in the way information is processed to generate the predictions. Therefore, the process is not entirely objective. So if we have a bias, like if I'm developing algorithm, I would have certain interest and biases which will impact in the predictions. While the cost of the supercomputing and high throughput screening has decreased appreciably over the past decade, establishing these pipelines still requires significant investments. Even though the computing techniques are cheaper, but identifying those compounds and then getting into the pipeline still needs significant investments. Ultimately, predictions made by computer have to be verified by a scientist to make sure they are valid. So we still need a medicinal chemistry involvement in the AI technology as well. Now, after the AI technology, I would call it biologics. So biologics is a newer trend in drug discovery. We are moving slowly from small molecules into the large molecules. So when you come to UK or Germany, most of the drug discovery is happening with large molecules. The small molecule companies are now less and less. Biologicals, these are the therapeutic products derived from biological sources. It can be vaccine, antitoxins, blood products, proteins, monoclonal antibodies, and some RNAs, mitochondria. These also is biologicals and nowadays used as medicines. Biologic versus small molecules. Only the difference is the complexity of the compound. So biologicals, including the biosimilars, are much more complex than small molecules. Small molecules are easy to handle, and we can easily design those small molecules and get into the in vitro, in vivo studies. When it comes to large molecules, the study is harder because it's not easy to design, number one. Number two, it should naturally available, and it should modify the disease state, and the animal models are not compatible with the large molecule studies. So we'll have to rely on mostly human in vitro studies, and in small cases into the human directly. Biosimilars, highly similar to the US licensed reference biological product, notwithstanding minor differences in clinically interactive compounds. Biosimilar is also another growing field. Like in India, we have generic drugs which are similar to the innovator products. They are small molecules, but the biosimilars are large molecules, generic drugs, you can say, but it's not exactly generic molecules. It's similar because no one cannot produce the same biological compound in a different lab. So it will be a similar compound. It can never be the same compound. That's why it's called biosimilar. So no clinically meaningful differences from the reference product in terms of the safety, purity, and potency. So what we can infer here is that the reference product and the generic product, the biosimilar, should at least match the safety, purity, and potency. We can't just do a bioequivalent study with this one because it never be equivalent, but at least it should produce the safety, purity, and potency. So we limited the clinical trials into safety and purity studies, and also the potency. So we'll say this is more or less to the innovator product, then that can come into the market. The third one is personalized medicine. So now we have seen the trends are AI and biological and gene therapy. These are the main three trends which are heavily focused in the Western countries for drug discovery. So gene therapy is mainly going towards the personalized medicine because people started realizing that one size fit to all is not happening. Many of the drugs, especially with cancer drugs, some of the patients are very well responding to the cancer drugs. For some of the patients are not responding to it. So the same drug given to the same disease condition to the same age group population, few of them will recover from the cancer completely, but few of them will die without any effect. So that's why people are started looking into the genes and then the effect. So personalized medicine is the future of medicine. So the basis for the personalized medicine is gene therapy, technique for correcting the defective genes. It is the process of inserting genes into the cells to treat diseases. Gene therapy is used to correct a deficient phenotype genes. So let me move this. Gene therapy approaches. There are a few approaches for gene therapy, as we know. Germline gene therapies. So these are mainly the genetic. The sperm and the eggs. They are the sperm germ cells. So are modified by the introduction of functional genes, which are integrated into their genomes. Change would be heritable because it's changed into the germ cells and would be passed on to the later generations as well. And another therapy is somatic. Somatic is nothing but no germ cells, the normal cells. The therapeutic genes are transferred into the somatic cells of a patient. Change will not be inherited by the patient's offspring. So it will not be transferred to their children or later generations. Gene therapy types: ex vivo gene therapy, transfer of therapeutic genes in cultured cells, which are then reintroduced into the patients. Example of that therapy is ADA deficiency. And in vivo gene therapy, a direct delivery of genes into the cells of a particular tissue is referred to as the in vivo gene therapy. Therapy, for example, is cystic fibrosis. So there are some vectors used for gene therapy. We can use viruses also in this case as vectors, the attenuated virus, obviously. So, by looking at the picture, you could know this is the retrovirus, one kind of corona is one kind of a retrovirus. Now, adenoviruses, adeno-associated viruses, and herpes simplex viruses also. Few DNA constructs, lipoplexes. Lipoplexes, nothing but nucleic forms which form a separate structure into the cells, and DNA molecular conjugates, and human artificial chromosomes. We can create artificial chromosomes as well. So there are some limitations to gene therapy as well. They are short-lived. The nature of gene therapy, immune response is heavy in this case, and problem with the viral vectors. Even though we use the attenuated virals, which could potentially cause problems, and multi-gene disorder. So when we change the gene in a human, there will be a disorder for multi-gene, and which could potentially affect your future generations as well. So these are the three main trends. I would say you can focus on. They have huge opportunities. There are opportunities in data science. There are opportunities in large molecules. So if you do your master's in, I'm not recommending, but if you do it in your pharmaceutical biotechnology, which has a greater scope in the Western countries, or you focus on your, there are plenty of fields, like you can focus on the clinical trial, medical affairs, regulatory affairs. These are the external fields to the our core R&D. So if you go on to that, it doesn't matter you are in small molecule, large molecule, or any other field. But when you are coming into the core R&D, if your interests are in core R&D, it's the growing fields or AI technology and large molecule gene therapy. So if you focus on those areas and trends, you will have more scope. This is about the trends and discovery. Thank you for listening, all of you. If you have any questions, you can raise.
Participants, you can post your questions, or you can feel free to ask any questions to the speaker. Meanwhile, Hari, thanks for the wonderful topic you have selected, and thank you for helping us to understand how the target selection from genomics, proteomics, and bioinformatics moves into lead discovery, then it moves into medicinal chemistry, in vitro, in vivo, and clinical trials. And thanks for the specialization which you have brought out about artificial intelligence, data science, biologics, biosimilars, and personal medicines as well. Okay, so as we are waiting for the question, let me start with some kind of general questions. Like you have already mentioned about pharmaceutical biotech and regulatory affairs, which would be suitable for the undergrad students here. As we are having a mix of students right from earlier semesters to higher semesters here, what kind of postgraduate courses are usually looked by the Western countries, which undergraduate student can opt?
Okay, so if they want to come into Western countries, or even though they are doing it in India?
Yeah, I mean in Western countries, because I understand that most of the students here are looking for one or the other country, especially in United States, Canada, or Australia.
Yeah. Okay, so there is a huge scope for artificial intelligence and biologicals and gene therapy as well. So they can focus on any postgraduate studies. Usually postgraduate in UK is just 1 year. I don't know about US condition. Within this 1 year, they are not hugely specified or specialized. So the people have interest while they are doing the bachelor's, and the bachelor's of 4 years, and they have the industrial placement as well. So by that time, they have the decision already made. In the Western countries, this is an advantage, you know, because you worked in Malaysia. People from bachelor, they can directly go into the PhD as well. So postgrad is not mandatory, but if at all, if anyone is interested, or is that the channel to come to UK or Western countries, I would prefer any masters in pharmacy, pharmaceutical sciences, or large molecule or biological or molecular pharmacology or pharmacokinetic or PK, clinical pharmacology. So there are some special focuses like clinical pharmacology, clinical trial, large molecule, and even PharmD courses are available for the master students. They can easily come into it.
That's nice. I think those information would be really helpful for our students as well. I'm waiting for the questions. If any of the participants have any questions, you can keep posting in the question and answer session, or you can straight away ask. If you could raise your hand, if you have any question, I can just open up for you.
Yeah, Dr. Hari, it was a very interesting session. Thanks for enlightening us the current trends which are happening in the UK. Can you throw some light upon health informatics as a program at post-graduation? What can be its scope? How they can be beneficial more towards industry?
Okay, so health informatics is another growing field. As I said, these all related to information and artificial intelligence. Like bioinformatics, it used to be a huge scope like 4 to 5 years ago. Now the AI is moving into in silico and other activities. The personalized medicine and pharmacoeconomics gaining more importance as well. So the health informatics, again, it's more related to the pharmacoeconomics. So it's combining the technology and the health industry, so which is also having a huge scope. But as I am coming from the core R&D, I don't have much experience into the pharmacoeconomics separately. But I would say I heard about it. Many students are doing it, and it has a good scope here, because reducing the cost of the national health services in UK is a important factor for the government, and they are investing much into personalized medicine, reducing the cost, and pharmacoeconomics as well. So they are a growing field as well, and huge investments are coming into it.
Yep.
Okay.
Thank you for that good insight about. Can you also give us some insights about students selecting programs of post-graduation at UK, basically, and followed by the job opportunities over there at UK? How do you feel?
Yeah, most of the students who are coming from India are just doing the MBA courses because that's cheaper compared to the core pharma courses. But it doesn't stop there. Mm-hmm. People can do the specialized pharma courses also. Now the Home Office has opened students from all over the world. What they have done is previously they stopped the post-study work visa. So now they open the post-study work visa, which means after their master's course of doing 1 year, they can have full work permit for another 2 years. So within that 2 years, if they can fetch a job in any of the pharma field or anywhere, they can then apply for a regular visa, work visa, because for the prior for getting a visa is we should have a job offer, but getting a job offer from India, it's difficult. So when they come here as a student and they work here with the post-study work visa for 2 years, they can obviously get into pharma industry. And then after joining in the pharma industry, which means they already have the job offer in their hand, which means they can go into the skilled migrant visa. So they can easily settle in UK. But for that, they can choose any course which offer the post-study work visa, because some of the courses are not offering the post-study work visa. So when they apply for these courses, they'll have to look into the universities. They easily provide the post-study work visa. And also they should have some connections to get into the pharma industry, because still it works the same way as it works in India. We should have the connections, the seniors or someone is working in the pharma industry, even though there are loads of agents working here, but the connection and their work experience help them. I would say, rather than coming directly into UK, if they have 1 or 2 experience in a specified field where they have the interest, for example, as you said, to pharmacoeconomics or the pharmacogenomics, whichever interest they have, if they have specific work experience and they come and do the post-graduation here, then with that experience, they can easily move into the industry in that particular field because they already know that field and they have the interest and they get into it, then they get the job offer, which means obviously they can get the skilled migrant visa and they can settle in UK.
Okay, Hari, I have a question
Yeah.
here from the earlier student. From the earlier semesters, I would say. The question is, so when you're discovering a new drug, what steps can be followed to know the source of the drug for a specific disease condition?
Source of a drug can be anything. Like there are 2 different approaches for drug discovery. In my company, we have the drug already. Like we are developing the drugs from cannabis. So we know what are the components in cannabis. Now we search for diseases which our drug can impact on. So we look into the molecular pharmacology. We study our compound structure. We modify the structure and we apply them into the molecular pharmacology. And then we work on neuroscience and we work on different disease areas. But the question is, how do we choose that compounds for a specific drug? If we know these are the unmet need means there are no drugs or there are less drugs for a particular disease, then we have to study the disease condition and the protein which affects that disease. Then we should have a random or we should have a computational experience of the drug molecule, the structure of the drug, which could fit into that target. So if we know, we can develop a library of compounds. It's not where we can find, we can synthesize them based on the structure. We know this is the functional group which fit into that specific target. So we can design that drug. We can design a number of drugs and we can choose those drugs. Then only that is called hit identification. So it's not where to find them. Of course, there are options. If you want, you can get it from the biologicals, or you can get it from the botanical sources, or you can synthesize them. But we should have a focus. It's objective of the company. Our company is a botanical company, so we source our drug from botanical. But some of the companies could be purely small molecule synthetic companies. They source from synthesis. And some of them for biologicals, they source from biologicals. So the disease condition is the same, but the sources are different. Based on our objective and our caliber, how to fit those 2, we can choose and source the drugs.
That's nice. That's nice, Hari. I think that will be really helpful for the students as well. And I would like to put another question to you regarding the skill sets. What is expected for an undergraduate? I would say if they are deciding to move abroad, or if they come back there to a Western country, let's say United States or Canada or wherever it be. What kind of skill sets are expected from an undergraduate or a postgrad student?
Okay, skill set is an important question. Number 1, they should have the flexibility of moving into the people. So when it comes to Western countries, not like India, they don't just focus only on the aspects of their knowledge. Of course, the knowledge and skills can be developed, and if they have the bachelor's degree, here in UK or any other Western countries, not many people do the graduation. They don't go to university. So if someone has done the university, obviously it's a consideration they are smart enough. So the smartness part is completed. When it comes to getting a job offer, they look into different skill opportunities. That's called soft skills. How do you have the flexibility? They will easily identify by interviewing what is the flexible nature of that person, adaptability, team working, how he handled the stress and how he handled the change in situation, and how he moves with the other colleagues. These all are the soft skills which are more important to fetch a job here, and the contacts, obviously. These are the necessary basic skills. But when you want to enter in a specific field, you should have the interest and deep knowledge of that one. For example, if some people may think, many of the people I know from Pune, that area, they would come and join in a regulatory affairs because they think it has a good scope. So you should have more knowledge in that area. Some people who would like to go into the intellectual property, so we should have in-depth knowledge in that and then focus on it. That are specific knowledges and skills related to that area. But apart from that, in general, they should have the Adaptability, flexibility, friendliness, easy to move, and risk taking, managing the stress. These kind of soft skills are more important to get into it.
Thank you, Hari. Thank you for adding those valuable points and making clear to our students that soft skills are more important to the being a content expert. And really, I think our university is definitely looking to the development of soft skills of our students here.
Yeah.
Before we end up session, I would like to have one last question. Can you please share your own experience as you moved to UK? You started your career as a bioanalytical scientist there, and right now you are in a lead position. So what are the challenges you faced? How do you overcome those things?
Okay, very good question. Actually, after having 4 years of experience in the bioanalytical field in India, I moved into UK. But it was not easy to get into the pharmaceutical industry. At that time, it was challenging because recession was there when I came to UK in 2010. That's not the reason. They don't consider the work experience and the university degree easily when we come to UK. So getting a job interview itself was difficult. So we need to modify our resume based on the resumes available in UK. You have to make it a little bit tricky and better. And then you have to, I have attended so many forums here in UK, not in India. They don't have specific interviews. Sometimes they will have open sessions. They just call people, because when you send the CVs to industry, they don't choose. They didn't choose, at least in my case, they didn't choose me. But I went to that open sessions. Then I talked to a few people there. Then they understand what's my skills. And when they have the confidence, number one, we should have better fluency. We should have the pleasing personality. As I said, they expect mainly the flexibility because they don't want to get someone to boss them. So, there are some hidden agenda. I don't want to talk in an open forum, but there are certain things which we need to be mindful of. So when they look into your personality and their approachability and your skills and experience, then they recruit into it. So that's how I got into the LDC. Worked there as a scientist. So wanted to grow in career, but it's not easy to grow within the same industry. It is the same in India also, but it's comparatively more difficult in Western countries. So I moved out of bioanalysis, gone into same analytical field, but in a pesticide analysis as a senior scientist, where I worked for 1 year. And after working there, they lost the contract. Then I was in a risk position to lose that job. Then I had to move back to bioanalysis, where I came back as a study manager. Because at that time, I had experience of having 4 years of bioanalytical experience in India, 3.5 years as a scientist in LGC, and 1 year as a senior scientist in Europe. So that helped me to get that position as a study manager. Once you got into the career ladder in any Western countries, if you have the brand of working in the industry, then it's easy to move on. The first job in a Western country is always difficult. So, and then I moved as a study manager, worked there for 2.5 years, then wanted a career progression again. It was not easy, again, promoted within the company. Even then it was like moving into study management. The senior study manager will do the same work of a study manager. So I wanted to move into program management, because until I come to the Envigo, the company called Envigo now, it's been taken over by LabCorp. As a study manager, I never worked in office-based environment. I was always in the lab, so I didn't know what the office-based work is. When I worked as a study manager, I started enjoying the office-based study management, interacting with the sponsors and clients, writing the reports, dealing with the scientists and scheduling them. These kind of administrative and management activities of the study was more impressive to me, more appealing to me than working in the lab. So I stopped working in the lab, completely moved into study management, which made me to go into the program management. Now I'm working as a discovery program lead, means a program manager in a discovery research. So I moved into GW Pharmaceutical as a program manager. What I'm doing here is there are several study managers, as I was in Envigo as a study manager. Now there are study managers, they work in specific studies. Some of them are pharmacological studies, some of them are synthetic chemistry studies, some of them are in the ADME study, in vitro studies. They all come into the program. They don't report to me directly. I'm not the manager of them, but I am responsible for the program. I'm handling a particular discovery compound to get into clinical. My responsibility is to take that compound from the hit identification to the clinical trials. So after clinical trial, it will be handled by a different program manager, because that is a huge volume of study when it comes to clinical trials. So there are more experienced program managers, but I am only program managing the discovery compounds. So in here, all the people who work in preclinical toxicology, pharmacology, synthetic chemistry, and the in vitro studies, they all come in my program and they report to me. It's like functions. They have their own line managers or their senior managers, but they will be reporting to them, but the program belongs to me. They can work in different programs also. The same people can work in different discovery molecule programs, but for my molecules, I am responsible. They have to report to me in terms of the work progression. So I keep monitor the timelines where the report is coming, and I'm responsible to send them to the regulatory affairs to file them to INDA, FDA, MHRA. And then I arrange the monthly meetings and I get the progress update. And I'll have to present this progress of that particular compound to my senior management, like the board of directors. I would be, because board of directors are focusing on the compounds, which other compounds are getting into clinical trials. That is money, in fact. Once you get the compound into clinical trial and that goes into commercial, that is what making the company money. So this is more important. So I will have often meeting with the board of directors to report my progress of the program. So that's called the program management in discovery. That's what I'm doing.
Okay, that's really interesting, and thanks for sharing your personal experience and how you moved up in your career. And definitely, I think this will be helpful for our participants today as well. Thank you so much, Harry. On that note, I think I got just popped one more question. Let me look into that. In gene therapy, the genes are inserted into the people from the family member. Where are the genes extracted from?
Okay, so it's not specifically extracted from any gene. It's like changing certain proteins. There are different techniques. As I said, there are vector microbes or the human being genome. There are germ cells that can be modified. 1 or 2 proteins can be modified. That does not necessarily come from the family member. It can come from the family member, or it can be synthesized protein, or it can be from the same person, or taken from a different tissue cells. And that gene can be used, or that can be developed in a vector, like the bacteria or virus. We can inflict that kind of a genes, because this gene might be missing in that person. That has to be developed from any microbe. Because the same family member, if you consider the family member, they all will have the same missing link, the same missing gene. If we want to replace that gene, have to develop that externally with any other vector microorganisms or in vitro human cells. So taking that gene and inserting into them, either external in vivo or in vitro, based on the therapy.
Okay, that's nice. That will be helpful, I think, for the participant who have asked the question. Any further question from the participants? I haven't seen any questions so far. So as there are no further questions, I think we can end our webinar session here. I would like to thank Hari for your time and insightful speech on drug discovery and what is the trend over Western countries and what's happening around the world. Thank you so much for this insightful talk for us. I'm also thanking our head of the institute, Professor Shivakumar, for giving us this opportunity and helping us to arrange a webinar talk on this day. Thank you so much, Professor. And I would like to thank the participants this afternoon. I believe you had a fruitful session and you must have gained some new information on drug discovery. I believe it will be helpful when you are choosing your career path. Thank you so much for all the participants and faculty members who have joined this session. Have a wonderful evening. Bye. Thank you.
Thank you, all of you. Thank you for Professor Shivakumar, and thank you, Kingston. And thank you, Geetham University, and all the participants for listening.
Yeah, thanks, Hari. I think we would look forward in future for your time and experience to share with us. It would be a great help to the faculty and also. Thank you so much.
It's my pleasure. Yeah. In future, if any other chances, I would be happy to take the sessions.
Thank you.
Okay. Thank you, Hari. Bye.
Bye.